So it's my pleasure to introduce Dr. Jason Buenrostro. Jason is a professor at the Department of Stem Cell and Regenerative Biology at Harvard University and a core member of the Broad Institute, where he serves as the director of the Epigenomics program and the co-director of the Gene Regulatory Observatory. Um, Jason's perhaps best known as the inventor of ATAC-seq, which is now widely adopted method for profiling chromatin accessibility genome-wide. His lab has since expanded this foundational technology into single-cell era, developing tools that illuminate how gene regulation governs cell identity, differentiation, and disease, with particular impact in cancer biology and hematopoiesis. His work has been recognized with a MacArthur Fellowship, the NIH Director's New Innovator Award, and a recognition among MIT Technology Review's 35 Innovators Under 35. I'll give the floor to Dr. Buenrostro.
Thank you. I appreciate that. It's great to be here and great to be with great company. I will begin. It's really great to talk to you. And I was mentioned, my lab's at the Broad and at Harvard, and what we've been really excited over the years is really thinking about how the epigenomes that live within us, right, of how they record the past and then predict our future health. So with that in mind, I think it's good to share with you how I think about the epigenome, and hopefully I can convince you that this is maybe a useful definition of what epigenetics is. But in my lab, we've been thinking about the epigenome as, of course, the things that happen to a cell's environment or to our personal environment, how that encodes into epigenetic changes onto our DNA, how that leads to changes in gene expression, and how that helps or causes cells to adapt, and therefore altering tissue function. And as we've thought about the epigenomes and our community has thought about epigenomes, we think quite a lot about all the changes that are occurring in development. We just heard about gastruloids from Jay, but also all the things that happen in aging. We know that upon age, cells are changing quite dramatically in their epigenetic code. It also happens in adulthood as we're exposed to our environment, as we eat, drink, smoke, stress, and breathe. And we also know that there's this kind of theory of the case that things that might happen to our parents or to parents in general may pass on that epigenetic information onto others. And the way, one kind of unifying way to think about this is the way we've been thinking about this in our group is that at some earlier life, I should say, you might have had an epigenetic altering event. That epigenetic altering event might persist in all of us to change who we are in some fundamental way. And then, of course, as exposures can sometimes do, right? Then some of these might lead to disease outcomes. So when you think about what kind of exposures we might be exposed to and we might find relevant, I kind of made a word cloud just to, think through the full diversity of things. Of course, inflammation, activity, aging, diet, so on and so forth. But all of these we know from public health research do quite a lot to imprint onto us and to drive disease pathology later in life. Okay, so there's going to be a few phases. First, we'll focus on the measurements, how we actually study epigenomes at this scale, and then we'll move into a case study that I think really exemplifies how we think about epigenetics in our group. So first and foremost, as some of you know, I've lost my name. I've now been referred to as the ATAC-seq guy. I do have a name, as Jason. But as some of you might be aware, we invented an assay a number of years ago now called ATAC-seq, where we could take unfixed chromatin, add in TN5 transposase, and then using sequencing actually read out where in the genome parts, things that are open and closed. And over the years, it's been a really kind of wild journey as I started my lab in about 2015. But we first demonstrated that single-cell ATAC-seq was possible alongside a work by Jay, showing that we can measure epigenomes in single cells. We've then worked to develop this field alongside commercial efforts and other people in our community to then adapt them to, say, droplet microfluidics. We've, of course, weren't really satisfied with just epigenomes. We wanted to link it with transcriptomes in order to understand how cells coordinate epigenomes and transcriptomes across, say, development. And then most recently, we've been really excited about the opportunity to build spatial epigenomics methods, not just thinking about cells within tissues, but also thinking about the nuclear and chromosomal architecture that lives within a single cell. So this was exemplified by our science paper just last year. And alongside, we've been building computational tools. We've been hard at work. You might, if you follow our work, have noticed that we try to build tools that allow us to not just measure these epigenomes, but to derive interpretation from them, where we can now link regions of open chromatin to gene expression, but also the expression of proteins like transcription factors and how they regulate specific genes to build out networks of how we think gene regulation is structured within a population of cells. But really, what I came here to tell you is that these tools exist now. They're really exciting, arguably. In our lab, we're generating about 10 million single-cell epigenomes per year. That number will, of course, change and very likely grow as the cost of sequencing keeps on dropping. And we now have these wonderful, wonderful AI tools that we've developed and that are in our community that decode this landscape. So what I thought is I'd start by kind of telling you how we're using these technologies in a few real world use cases. So one of the questions we've been asking in our lab for quite some time is just this broad question of how do signaling pathways cooperate to drive a change in lineage or a change in cell state or identity. And as we think about this problem, of course, you could think about it in different ways. We've been thinking about it in our group as the physical interaction and the cooperation of transcription factors on DNA. There's plenty of examples out in the literature, and none of these are kind of any modern revelations. This is, of course, well known that transcription factors co-bind to drive gene expression. So here's just a famous example of three proteins, phos, jun, and NFAT, that come together to drive T cell activation and serve as a rheostat to lead to T cell exhaustion. So in order to tackle this broader problem, we thought between Jan Hu, graduate student in my lab, who now doing a postdoc, Max Horlbeck, now running his own lab, and Richard Zhang, still in my group, we thought to create tools that actually unravel this molecular complexity and protein interactions on DNA. So to do that, we really wanted to come back to some of the roots and accessibility mapping. So this is culminated in a paper in Nature just last year, where we wanted to use ATAC-seq data to bind where transcription factors are by identifying where there might be footprints or gaps in cutting of the TN5 enzyme. So for many of you, you might be more used to looking at epigenomics data looks like this. So if you just scan across, you'll find a peak. But in ATAC-seq, if you just zoom in to a ATAC-seq peak and start to quantify the individual cut sites of TN5, you'll find that there's different intensities and propensities of cutting at different locations. And you'll find these dips and these valleys, right? Some of these might be due to just air or noise or bias in the TN5 cutting frequencies. But some we would anticipate are due to these macro complexes that might form on DNA called these transcription factors that might form something called a footprint. So I'm going to skip through a whole lot of kind of trial and error statistics and AI/ML, and just kind of give you the highlights of why we think this is super exciting in terms of advancing our understanding of gene regulation. So one of the core things we figured out in this work is that we can convert these sorts of plots of individual cutting frequencies to now, these are log 10 p-values, actually is footprint scores. But what you'll find here in this example is here on the x-axis is a genomic position. And here on the y-axis, we're looking at footprint size. So these are like the taller things are on this plot, the bigger the protein is, small versus large. And we're converting a kind of 1D sequence, applying a statistical filter, accounting for sequence bias and all that. And then now creating an image for the genome that we can now start to interpret. So in this particular plot, we seem to find three proteins bound to DNA. Here in the middle is, because we know what it is, I'll just tell you what it is. It's CTCF. And then here at the flanks are two nucleosomes that flank that particular binding event. So we were really excited. We were finding these beautiful TF binding events inside cells, but one of the things we really struggled with is interpreting these images, right? These images we're not used to, in the same way we might recognize a face, we're not really used to looking at kind of these sorts of images of the genome. So what we decided to do is to move into deep learning, inspired by work by Ashok and others, where we can now take DNA sequence and use it to predict these multi-scale footprints in effort to connect the DNA sequence to the footprints we're seeing. So again, I'm skipping through a lot. Hopefully that's okay. But what we're able to do is use this method to predict the binding of transcription factors. So we were really astounded by how specific these predictions were, much better than just motif matching, much better than traditional footprinting methods, and now letting us get to a resolution that we think is quite good for actual use in terms of predicting where transcription factors are. And really interestingly, it didn't just predict the transcription factors we knew. It also allowed us to discover new transcription factor complexes we didn't know were there. So here I'm just showing 3 de novo motifs that we couldn't easily identify their source, but we found them. And here in the middle was actually one we were able to annotate because this turned out to be 2 individual SOX binding events. You see SOX here, just in the reverse orientation. And you can actually make out pretty nicely that there are these 2 individual footprints of 2 transcription factors that now tell us that there are 2 individual binding events in that particular window. So using these methods, we've been applying it in the wild. One place we applied it in the paper in collaboration with our department chair, Amy Wagers, is to understand how hematopoietic stem cells might change upon age. And as you might know, hematopoietic stem cells and many stem cells in our body are get quite exhausted upon aging, and in this particular case, they'll actually skew in their lineage to then drive inflammation throughout the body. So we applied our tool, single-cell ATAC-seq, onto these cells, and then we're now keen to understand the transcriptional regulation associated with aging. So as we looked through this work, we actually found a lot of transcription factor motif sequences that we knew to be dysregulated in aging. So as you look up, the ones in red, these are the ones that are aging associated. The ones in blue, these are the ones that are aging depleted. And we found things like FOS, RUNX, GATA, so on and so forth, were up in aging as expected, as well as many novel composite motifs, here just labeled as motif number 8, 10, 18, 27, and so on and so forth. And of course, we're interested in the novel. We weren't expecting to see these things, therefore we dug into them quite a good deal more deeply. And what we actually found is many of these novel motifs were actually well explained by composites of 2 known things. Specifically, some of the top ranked hits we were finding were composites of the 2 transcription factors ETS, shown here, RUNX, at different orientation, spacings, and structure. And as we looked at all the motifs, we found this to be a recurring pattern, where many of the differential motifs we're finding in aging were actually different directions. So each arrow is, of course, denoting the direction of the sequence. And when you put them all together, we found that they were actually leading to a really strong signal in aging, because, of course, every one of these instances is diluting your statistical power. But when you bring them all together as a unit, you, of course, we're seeing quite more significant effect. So what we're left with in the paper is how do we validate an interaction among 2 transcription factors on the genome, specifically in aging HSCs, where we know doing ChIP-seq is just near impossible to do. So what we decided to do is come back to structure. So we were lucky enough to know and to discover that the crystal structure for RUNX and ETS had already been determined, showing that very directly that there was a functional interaction between RUNX and ETS, shown here. And interestingly enough, as we started giving AlphaFold 3 deep learning model that is able to resolve protein-protein DNA interactions, we actually found that there's quite a lot of evidence that we can use as support that these AlphaFold 3 models were able to predict interactions among ETS and RUNX across these different motif spacings and structures. And what we found as a unifying theme is that there's actually, if you can see here, this is long flexible linker between RUNX1 and this EID domain. The EID domain is known to activate ETS and to cause ETS to be transcriptionally active, explaining this flexible motif syntax that we see. So I use this a bit of tongue in cheek, but it looks like a deep learning model validated a deep learning model that built some comfort and some excitement for us that we're on the right path. And as we've been going across these different studies, we've actually found that TF-TF interactions are no exception. We're finding, as many others have found in the field before, we're finding tons of examples of two transcription factors coming together to form what we suppose to be a more stable interaction, but also integrating signaling within the cell to lead to very specific gene expression outcomes. And what I showed you was HSC aging. What we'll talk about next is some very recent work where we showed that interaction of transcription factors leads to inflammatory memory. And what I won't show today, but I hope to put as a preprint very, very soon, is we actually find a novel interaction associated with T cell activation and exhaustion that we're excited to report soon. So what we've been thinking about in the lab, motivated by this work, is can we discover TF interactions that drive pathogenic cell states in aging and disease? So we've been, again, applying this to as many contexts as we can think of. And to begin this discussion, I want to come back to this view, which I showed you again. And we're really, in the lab, we've been keen for years now to understand how exposures, of course, lead to disease. So one thing we've been really curious about in the lab for quite some time is inflammation. We all know inflammation. You can feel it as you get a rash, right? You can know that it happens inside your body and outside. But what you might not know is that it's actually a major risk factor for cancer in that same tissue. So the more inflammation bouts that you get in the tissue, the more likely it is to predispose you and give you high risk for cancer. And this association is well known in the clinic and known by many. So I'll just highlight when it was some of the earliest work that describes it. We're really interested in this to dive into the connection between colitis. Now we're thinking in the colon, thinking about damage to the colon that's leading to inflammation locally in the colon, and association with colorectal cancer. So in this New England Journal of Medicine paper in 1990, it was actually shown that the duration of ulcerative colitis directly scaled with the colon cancer incidence risk. And these effect sizes are quite significant and, of course, really kind of induce risk to individuals with this disease. So Surya Naharaja in my lab, a postdoctoral fellow, came into my lab and was really interested in tackling this question. So what we decided to do is to set up a mouse model in the lab where we induce colonic inflammation by giving them colitis with low-dose DSS. And then we chased, right? We then observed what happens to the tissue over time. So because I'm going to move a little quickly, again, I'll give you the highlights of the work. It just got published just a week or so ago. So of course, you called this kind of a flash talk for that paper. So we had several findings. One finding is that in stem cells in the colon, that stem cells remember colitis long after you remove this external stimulus that induces inflammation. So I'll just show you what that looks like, and we have lots of data in the paper you can look at. But here in the purple line, we're actually tracking the FOS protein that we know is activated in response to inflammation. And of course, you see it go up in inflammation. And after you withdraw the inflammatory stimuli, you see within 50 days, you see no levels of inflammatory signals. And we also saw no morphological changes at that point, and things were kind of crystal clear. But if you looked at the accessibility around the transcription factor AP1, you see, again, a peak in activity soon after the highest points of AP1 protein. And then you see it persist long after the stimulus, 100 days after the stimulus is withdrawn. Motivated by that, kind of skipping to the punchline, we found that this memory of inflammation actually induced high cancer risk. So when we then added a known oncogenic mutation, knockout to APC, we see that we not only were able to induce tumors, we're actually able to induce much larger, much more aggressive tumors in the mice that have otherwise fully recovered from this inflammatory bout, suggesting there's long-term memory of this inflammatory bout within mice. And finding number 3 is that we actually went back and looked and found that a lot of these memory events coincided with changes in DNA methylation. And what I'm not showing on this slide is that inhibiting AP1, we inhibited it chemically, does not erase this memory. So here at the bottom, I'm just going to show you really briefly differences by chromatin accessibility by ATAC-seq. Every dot is a region of the genome that's open to close. And this is, of course, colitis versus control. And then here on the y-axis is changes in methylation at these loci. And you can see that the R value is actually not so bad. Whenever we see a change in accessibility, it tends to coincide quite significantly with changes in DNA methylation at these loci. And that's true also when you look at specific genes. Here's one specific gene where we can actually resolve that there are individual CPGs, single individual methylation events here at the bottom, that seem to directly coincide with this change in accessibility. So the reason I bring this up for today's talk is that I'm going to now transition to kind of the meaty part of the paper, which is we wanted to ask how memory was imprinted. We see that this memory is there. We associate it and implicate it as part of the pathology of cancer risk, but how is it actually imprinted? So we went back to our newfound tool using AI to find these sequence composites. We found that there were very interesting composites, including this really interesting one by Fox proteins and also AP1, shown here. And that particular Fox AP1 protein seemed to have a pretty profound, by modeling and by prediction, effect on accessibility. So by altering the proportion of that motif inside a part of the genome, you can actually of course, induce the binding of the transcription factor, but that would lead to the dramatic loss of a nucleosome just right there, suggesting it's affecting chromatin structure quite specifically. So in order to validate this, Conrad Huck in the lab, together with Surya, worked together to develop a biochemical assay, which I think is really neat. What we did is we took the top 63 genomic loci in the genome where we saw this memory effect. We then PCR'd those regions up. We purified AP1 and Fox, and now biochemically brought them together. We then put in TN5, all outside of cells, all in vitro, to then see the footprints that emerged as a response. And the TLDR is when we looked at different regions of the genome, all 63 regions told the story, but I'm telling a summary now, is that Fox P1 alone did not really bind these regions. FOS in June, AP1 barely bound those regions, but when we added all three transcription factors biochemically, we saw a massive increase in the binding of AP1, stabilizer mediated by the binding in this case of FOXP1. It's no surprise that AlphaFold supported this result. So you actually see FOXP1 kind of folding over, kind of holding on to the chopsticks-like structure of AP1. And then one thing that was really fascinating, which we didn't really highlight so well in the paper, but I'm highlighting for you, is that FOXP1 can bind directly with June to stabilize it to DNA. And we actually bared that out with the biochemistry as well, suggesting that this might actually serve, this is just me spitballing now, serve as a catch mechanism to then kind of recruit FOS more strongly and more effectively to sites. And strong thank you to Jay, who actually put together News and Views. I'm just showing it because I had to give him credit for this wonderful News and Views associated with the paper. But in summary, we actually find that inflammatory response will lead to epigenetic memory, and then that those cells that remember are now primed for cancer through this mechanism of AP1 and FOX co-binding and the alterations of accessibility and methylation. So as follow-up to that work, we're looking in humans, building tools for diagnostics, interventions. Of course, this is not going to be a one-year project. We're really motivated by these signals in order to then advance our ability to track risk within human populations. So now I'll close with just saying a few remarks. Just one that I thought I'd share with you is that there's our work using AI. There's also Alpha Genome. There's also Anshul's work, and many others now are contributing using single cell and bulk ATAC-seq data. But there's major opportunities for epigenomics leveraging AI. We're discovering so much. It's a really wonderful time to be in this field. And then the second thing I wanted to mention is that we and others like Elaine Fuchs have now started to find more and more examples. Arjun Raj is also working towards this direction, that cells accumulate inflammatory memories causing adaptive, in the case of Elaine, showing a wound healing response. In the case of our work, I'm showing a maladaptive effect on cancer risk. And then maybe I'll mention this as a last point because I have your attention, is that we're actively recruiting to work on these sorts of projects, and it'd be great to hear from some of you if anybody's interested in opportunities in the group. So with that, I'll just end now and say thank you to the lab, whose, of course, contributions have been felt throughout all the work that we do. Thank you to Surya. Thank you to the three first authors that led the footprinting work, to the collaborators that made it possible, institutions, and the funding support that provide the community that we get to do this wonderful science with. So with that, I'll pause, and I'm looking forward to that discussion.
Do you know the different motive orientation spacings that lead to different effects on expression of the putative target genes?
Yeah, so just to kind of re-ask the question, the motifs can be in this orientation, that orientation, also at different spacings. And in the first earlier work, we didn't pay much attention to that. We just kind of were like, oh, that's just must be like this flexible linker must accommodate quite a lot. But in work I didn't show today, and work we've been doing in the lab, we actually find that they do drive distinct sets of genes, which is quite crazy. And we're not really sure if it's like these two proteins in different orientations will recruit different cofactors, because you can imagine there being a different interface for a cofactor, or if it's different proteins that are looking at that composite motif and then binding in different ways. This relates to another question here. As you might know, there are a few different RUNXs, many different ETs, and lots of family members of AP1. In Surya's paper, the Naharaj et al. Just came out, we actually looked at two FOX proteins based on which ones we thought were most dysregulated in colitis. And they both co-bound with AP1, but their co-binding affinity was quite different. And if you look, you'll find that there's actually quite interesting differences across the two. So I think I answered both questions there in one go, which is the picture is quite complex, but I do think that these different orientations and spacings are doing different things, recruiting different things, and also using TF family members in different ways.
How sensitive is print to sequencing depths? So there are labs that might have modest research budgets. Do you think one can apply print to single cell data from sparse data sets?
Yeah, I think how much was it? It was 5 million reads per cell. But it might also depend quite a bit on the applications one is most interested in. What we found is fine tuning, like you can build a big model with say other data, public data, and then you can fine tune it for your particular use case. The fine tuning usually needs less um, reads, and then as you could also imagine, it might really depend on like how highly resolved you want to actually define a certain interaction. So I'm always hesitant to be like, oh, this is the thing you need, because of course the more data you have, the better the models will do. But I would say after 5 million reads, you get start to see the diminishing returns, where at more than 5 million reads, you get more, but it returns less.